Who are you, actually? What are all those little chromosomes in your body really up to? What about this guy Mendel and his pea plants? Have scientists truly found a new species of fire-breathing tree monkeys in the South American rainforest? I can't answer these questions, but this show can.
The cell cycle and mitosis.
Where do cells come from?
Well, they come from other cells.
Cells divide. But a cell doesn't just click its heel 3 times and poof, it's divided. No, no, no. There's a path the cell must follow. We call this path to cell division the cell cycle.
Basically, one cell splits and becomes 2 new cells that are identical to the original. That's cell division in a nutshell. One cell splits and becomes 2 new cells identical to the original.
Madame Vocabulary at your service. Your future is unclear. Here are some definitions to help you navigate. Don't worry about memorizing them now, traveler. Just get used to them, like a new pair of blue jeans.
Mitosis is the process in which the nucleus of a eukaryotic cell divides into 2 identical daughter nuclei. The nuclear membrane, or nuclear envelope, is the layer that encloses the nucleus in eukaryotic cells. A nucleolus is a structure in the nucleus that is formed from dense areas of chromosomes and that is active in making ribosomes. Nucleoplasm is the liquidy substance that surrounds the other stuff in the nucleus, kind of like the squishy cytoplasm in the cell. Ribosomal RNA, rRNA, is the most common type of RNA, ribonucleic acid. It is developed in the nucleolus.
Stay with me, travelers. The transmissions are flowing strong. Just let them wash over you.
Chromatin is the stuff that makes up chromosomes. It's a mixture of DNA and proteins. DNA, let's not forget, is a nucleic acid in the nucleus that contains genetic information or instructions for how an organism will develop. Chromatids are duplicated chromosomes, like identical twins, that are attached to each other. A centromere is the center point where 2 sister chromatids are joined. A spindle, or mitotic spindle, is a jellyfish-like structure that forms in the cell to assist in cell division. Cytokinesis is the dividing of the cytoplasm immediately after mitosis in order to form 2 separate daughter cells.
All right, already. Let's learn about the cell cycle. In particular, that majorly important part of the cell cycle, mitosis. But before we do that, let's take a look at a typical cell. And let's give special attention to the nucleus, since that's where mitosis goes down.
You got it. Here's a typical cell. Here's the nucleus inside, protected by the nuclear membrane. Now let's get closer and see what's inside the nucleus, because it's some pretty important stuff. There's the nucleolus, where ribosomal RNA is stored. And see these patches? They're chromatin, where DNA is stored. And here's the nucleoplasm all around.
As you can see, the chromatin stuff is all spread out in the nucleus. Eventually, it will sort of condense into chromosomes, but not yet because the cell division process hasn't started. Right now, the cell's just chilling out and being its normal self. In this state before cell division, it's considered to be 2N.
Hey, watch it with your language. What the heck is 2N supposed to mean?
Okay. It just means that sexually reproducing organisms, like humans and animals, have 2 sets of chromosomes in each of their cells. 1 set from mom and 1 set from dad. So 2N just means 2 sets. All humans have 23 chromosomes per single set. So 2N is 2 sets of 23 chromosomes each, which is 46 chromosomes per cell. Easy.
Okay, let's get this cell cycle started already.
Here's the cell cycle. There's interphase, which has 3 phases, G1, S, and G2, and there's this region of mitosis, which consists of prophase, metaphase, anaphase, and telophase. There's also this thing called cytokinesis that occurs in telophase. And then the cycle starts all over again with interphase. That's a lot of labels. What does this all look like? Luckily, we've got a little home video here of cell division.
That looks like synchronized swimming. Let's understand what all those moves mean.
First up is interphase, the first phase in the cell cycle. Interestingly, interphase has 3 phases in itself, G1, S, and G2.
Interphase is all about growth and duplication. You can think of G1 and G2 as growth 1 and growth 2, and S as synthesis. Synthesis just means to make. In this case, it's when the cell makes copies or duplications of its chromosomes to prepare for division. The reason for this is simple. Since the cell is going to be splitting from one cell into 2 cells, and since both of these daughter cells are exactly the same as the original, it's going to need 2 sets of those original chromosomes. You guessed it, in interphase, the cell duplicates its chromosome count from 2N to 4N. Let's see what it looks like.
So, during G1, the cell grows. Also, the organelles, such as the mitochondria and the endoplasmic reticulum, are duplicated.
The S phase. In the S phase, the DNA in the chromatin is duplicated, so the cell is now 4N. You're probably thinking that you thought we said 4N referred to the chromosome count. Well, it does. Just remember that the chromatin hasn't coiled up into the shape of chromosomes yet, so we can't really see them at this point. Right now, it's just a big mess of chromatin. The important thing is that the stuff has been duplicated. There are now 4 sets of chromosomes, 2 from your mom, 2 from your dad.
G2, the cell, which is now 4N, grows even more. Grow, cell, grow! Gross.
Interphase, G1, S, and G2. Growth, duplication of the genetic material, and more growth.
Enough of interphase. Now onto the main event of cell division, mitosis. Mitosis has 4 phases. Let's see. Prophase, metaphase, anaphase, telophase. Let's check them out.
So mitosis is the actual splitting of the cell, specifically the nucleus, the cell's main headquarters. Interphase has prepared the cell for mitosis by duplicating the chromosomes count. Our single cell has gone from 2N to 4N. Now, when the cell splits, each daughter cell will be 2N, just like the original was before all this madness began. Get it? Each daughter cell will have the same genetic blueprint as the original.
Back to our nucleus to check out prophase, the first phase of mitosis. In prophase, the nuclear envelope enclosing the nucleus disappears. And the nucleoli disappear too. And the chromatin develop into distinct chromosomes.
Yeah! That's what I'm talking about. Yeah!
But remember, the chromosomes have been duplicated during interphase, so what we see is a slew of identical chromosomes. The identical chromosomes are called chromatids, and each identical pair are attached to each other at a point in the center called the centromere.
Just think about it like a pair of identical Siamese twins, joined at the hip. Instead of saying, look, there's 2 people who are exactly the same stuck together. We want to be concise, so we give their stuck-together identicalness a name. Siamese twins. It's the same with chromosomes. We call identical chromosomes that are attached to each other chromatids.
Let's watch prophase again. So the nucleus and the nucleoli have disappeared, and the chromatin has formed into chromosomes made up of identical chromatids. One more thing that happens in prophase is that this thing, called the mitotic spindle, begins to form outside of the nucleus. It's kind of weird, like this big alien jellyfish hovering over the nucleus. Creepy.
It gets creepier. Let's watch that one more time. Since the nuclear membrane has disappeared, the alien jellyfish tentacles of the mitotic spindle get inside and attach themselves to each of the centromeres. Yikes! The centromeres, remember, are the points where the twin chromatids are attached at the hip.
Okay, next up, metaphase, the second phase of mitosis. With the spindle fibers attached at the midpoints of the chromatids, the chromosomes now line up along the equatorial plane of the cell. Looks like they're getting ready for something to happen. I wonder what it'll be.
Moving right along to anaphase, the third phase of mitosis. Now those creepy spindle fibers pull the chromosomes apart at the centromeres. That is, the chromatids get pulled apart, and they go their separate ways. No longer Siamese twins. One heads to this side of the cell,
One heads to the other side of the cell. So long, farewell, each ready to be a single chromosome all by itself inside its own cell.
Telophase. All right, things are looking good, and it's time to really make two separate cells. So guess what happens next? A nuclear envelope forms around each set of the duplicated chromosomes, so they've now got their very own nuclei to live in, and the cell starts to divide.
Now that last part there, the divvying up of the cytoplasm evenly between the two cells and the final splitting of the cell, is called cytokinesis. Cytokinesis starts in telophase and finishes off the cell division process.
There's one thing you should know about cytokinesis. The thing is that plant cells and animal cells do it a little bit differently. Check it out. In a plant cell, a cell plate forms down the middle and separates everything into two cells. Pretty straight up. An animal cell's got a little more style. Animal cells divide along a cleavage furrow. That just means they pinch together and then pull apart.
Okay, folks, that's the end of mitosis. We've ended up with two new cells, each exact genetic copies of the original cell we started out with.
Don't forget, mitosis is the cell division part of the cell cycle, and it's mainly about the action in the nucleus. Mitosis happens in all the cells of your body, except the sex cells.
Before we finish, let's recap the parts of the cell cycle. Here's a handy way to remember them. Interesting pro met Anna on the telephone. That will help remember interphase and the 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. It's silly, yes, but so helpful. Interesting pro met Anna on the telephone. Interphase, prophase, metaphase, anaphase, telophase. Don't forget that interphase has 3 phases in itself: G1, S, and G2. It really is quite interesting.
Hey, why not add cytokinesis in there as well? How about interesting pro met Anna on the telephone? Psych! Psych will remind us of cytokinesis. See ya!
Meiosis.
Meiosis is a type of cell division that occurs with only one type of cell, the sex cells. Meiosis is kind of like mitosis, the cell division that occurs with all of your other cells, but there are a few big differences. So listen up.
Greetings, travelers! Your future is becoming ever more clear with much interesting vocabulary in store.
Meiosis is the process by which sex cells divide to produce daughter cells with half the number of chromosomes as the original. Gametes are sex cells or reproductive cells, sperm in males and eggs in females. A diploid cell is a cell that contains 2 sets of chromosomes. A haploid cell is a cell that contains a single set of chromosomes. A gene is a chunk of DNA that contains instructions for a particular genetic feature. The locus is the specific location on a chromosome where a gene is found. Homologous chromosomes are pairs of corresponding chromosomes from a sperm and egg cell that contain the same gene. Crossing over is when DNA molecules from non-sister chromatids swap genetic information during prophase 1.
Before we jump into the phases of meiosis, we need to take a moment to distinguish between asexual and sexual reproduction. In asexual reproduction, there is only one parent that makes an exact copy of its genes and passes the copy to its offspring. So the offspring produced by asexual reproduction are exact copies of their parent. Pretty boring.
Sexual reproduction is where things get interesting. In sexual reproduction, you have 2 parents that combine their genes in all different ways to produce offspring that are different from them. Since each combination is different, each offspring comes out differently. Humans, for example, produced by sexual reproduction. You are different from both your parents and different from your brothers and sisters. That's what makes life interesting.
In sexual reproduction, parents pass on genetic information to their offspring. You may have noticed that some things about you are the same as your parents and some things are different. How does that happen? Well, it has everything to do with genes. Genes are like little built-in instructions that determine how parts of us turn out. We have all different kinds of genes that make us who we are. A gene for eye color, a gene for hair color, even for tongue rolling.
Genes, the very basis for our individuality. And also those strange similarities we share with members of our family. See this hair? My father was bald. My brother is bald. Not I. Not I!
Now it's time to look into this business. Meiosis is a really important part of sexual reproduction. Remember, it's a type of cell division that only goes on with the sex cells. It is super important to understand that meiosis starts with one cell, then creates 4 daughter cells that are not identical to the original cell. Sounds juicy.
Before the process of meiosis starts, the cell is just chilling out like a regular cell. It's 2N, meaning it has 2 sets of chromosomes, one from mom and one from dad, just like every other cell. But guess what? The reproductive cells, or sex cells, are the only cells in the body that need to go down to just one set, or a single N. We'll talk about why this is later. But for now, remember that cells with 2 sets, or 2N, are called diploid, and cells with 1 set, or N, are called haploid. Think half for haploid. Meiosis is about going from a diploid cell to a haploid cell.
Let's get our bearings. We said that meiosis occurs in 2 rounds, which each contains 4 phases. The first round, meiosis 1, has prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2, the second round, has prophase 2, metaphase 2, anaphase 2, and telophase 2. What a trip! Let's check out a little home video of this process.
Before we watch, let's just take note of one thing that's going on here. We know that a human cell has 46 chromosomes. That's 2 sets of 23 chromosomes each, right? Well, that's sort of a lot to draw for our models, like putting 70 candles on your grandma's birthday cake. So we're just going to use 6 chromosomes with 2 sets of 3 to represent a full cell. The set from mom is pink, and the set from dad is blue. Okay, hit it.
Just like in mitosis, before meiosis even starts, there is a period called interphase, when the chromosomes get duplicated into 2 sister chromatids, which are like Siamese twins attached at the hip. Now pay attention to this. Although the DNA has been doubled, the chromosome count stays the same. That's because 2 chromatids are still only considered 1 chromosome. I repeat, even though the amount of DNA has doubled, the cell is still considered to be 2N, not 4N like in mitosis.
2 chromatids stuck together are still only considered 1 chromosome. It's wrong. It's not fair. But you may as well accept it. They must be separated to count as 2 chromosomes.
The first phase of meiosis 1 is called prophase 1. At this point, the cell is 2N, diploid, with 2 identical chromatids per chromosome. Now check this out. In meiosis, the chromosomes don't just come together, they also line up in pairs called homologous pairs, or homologous chromosomes.
Let's make sure we understand what homologous chromosomes are. It's really simple. Cells have 2 sets of chromosomes, one from mom and one from dad, right? So, the set from your dad has a chromosome that contains a gene for eye color, and so does the set from your mom, right? Well, these 2 come together as a pair so they can negotiate which eye color their child will have. The 2 chromosomes with the genes instructing for hair color come together, and for the length of your toes, too, and so on. That's what we mean by homologous chromosomes.
We're going to represent homologous chromosomes here by size. The big pink chromosome from your mom lines up with the big blue chromosome from your dad, and the small pink with the small blue.
The pair of joined homologous chromosomes is called a tetrad, which means a group of 4.
Don't forget that each chromosome has just been duplicated and contains 2 chromatids. The 2 from your mom are identical, and so they're both arguing for blue eye color, while the 2 from your dad are identical, and they're both arguing for brown eye color. That's 4 chromatids for each tetrad, and they're all talking about the same feature, in this case, eye color.
The next thing that happens in prophase 1 is really, really important, so listen up. It's called crossing over. Let's see that video again.
When our homologous chromosomes are in tetrad formation, they do more than just line up. See how some of them get crisscrossed and the colors get a little mix-matched? That's called crossing over. One of your mom's chromatids is like, okay, I'll consider brown eye color, and one of your dad's is like, fine, I'll consider blue. Gosh. And so they both mix their ideas, aka genetic instructions, together a bit. This is how you end up carrying different combinations of genetic information from your parents.
At the end of prophase 1, the nuclear envelope and the nucleoli begin to disappear, similar to mitosis. Now, in metaphase 1, they're all gone, and the tie-dyed tetrads line up along the equator of the cell, getting ready for some action.
In anaphase 1, the homologous chromosomes separate, guided apart by those jellyfish-like spindle fibers. The chromosomes have had their conversations, and now they go their separate ways. But remember, they still have their chromatids, which have not been separated yet. But guess what? The chromatids are not all identical anymore. Everything is all mixed up from the idea swap. Although some of the chromatids have stayed the same, some of them are walking away from this encounter quite changed. It's like returning home from a summer at camp. Maybe you've met a boy or girl. Maybe you've learned archery and you're changed. In telophase 1, as in mitosis, the nuclear membrane reforms. Cytokinesis occurs and pinches the cell into two daughter cells. So now, the daughter cells are 1N, haploids. They have one set of chromosomes each, but each set is a random mixture of mom's info and dad's.
Okay, so we've gone from a diploid to a haploid cell, from 2N to 1N. Aren't we done?
Nope. Look, the chromosomes are still made up of 2 chromatids each. They still contain twice the DNA, so we gotta pull them apart. And now meiosis 2, which consists of prophase 2, metaphase 2, anaphase 2, and telophase 2.
Okay, we've got 2 cells at the start of meiosis 2, remember? In prophase 2, the nuclear membrane disintegrates again. In metaphase 2, the chromosomes line up on the equator of the cell once again, and the spindle fibers connect to the chromosomes at the centromeres. In anaphase 2, the spindle fibers pull apart the chromatids, and they each head to a different end of the cell. They are now considered one chromosome, like one whole person. In telophase 2, the 2 daughter cells split apart via cytokinesis, and we've got 4 new cells.
These are gamete cells, or sex cells, otherwise known as sperm and egg cells. Notice that each of these cells has only 3 chromosomes, whereas the original cell had a total of 6 chromosomes. Gamete cells are haploid, or half the number of the original. That's 1N.
These guys have their own bank accounts now, and their own email addresses, and their own votes.
Yeah, we do. That's right. Woo!
So back to that question, why is meiosis necessary? Why go from diploid to haploid? Gametes, that is sperm or ova, need to only have one set of chromosomes so that they can eventually join forces with another gamete to sexually reproduce. That's what meiosis is for, reducing the reproductive cells from 2 sets of chromosomes, or 2N, to 1 set, or N, so that when they reproduce, the chromosome count becomes 2N again.
Right. And so meiosis occurs somewhere between the time of your beginning as a fertilized egg in your mother's body and you're becoming a sexually mature adult, ready to reproduce. Well, that makes sense.
And don't forget, because of the recombination of homologous chromosomes, you know, when your mom's chromosomes converses with your dad's chromosomes and then get all mixed up, because of that process, you get all different combinations of features, which is why your children will be different from you. Because of meiosis that occurred in your parents, you are different from them and from your siblings. Wow, without sexual reproduction and meiosis, life would be so boring.
Before we go, let's take a second to compare mitosis and meiosis. Both processes require the initial duplication of DNA. The main difference is that mitosis starts with one diploid cell and uses one division process to create 2 identical diploid cells, while meiosis starts with one diploid cell and uses 2 division processes to make 4 haploid daughter cells.
And of course, meiosis sounds a bit like meow.
Mendel's laws of inheritance.
Let's find out about this guy Mendel and his laws of inheritance. Shall we?
Gregor Mendel, a monk who lived in the 1800s, was one of the first to really study genes, inheritance, and those kind of things. He did a lot of work with pea plants, like these guys. Basically, he did a lot of crossbreeding with his pea plants to explore different traits among the species, such as height and color.
Madame Vocabulary here with more transmissions coming through of important hereditary and inheritance vocabulary.
A trait is a characteristic of an organism, like hair color for humans or height for pea plants. A gene is a chunk of DNA that contains instructions for a particular genetic feature or trait. An allele is a version of a particular gene, like blue eyes or brown eyes. A gamete is a sex cell, the female egg, the male sperm. Fertilization is when two gametes, a sperm cell and an egg cell, fuse together to create a zygote. A zygote is a fertilized egg. It contains two sets of chromosomes, one from each of two gametes.
Now before we start, let's make sure we are really clear about what an allele is. An allele is the form the gene takes. For example, you have a gene for eye color, meaning your body has these instructions to give you an eye color. But what color will you get? Blue, brown, green? These versions of the genes are alleles. My allele for eye color is brown, see? How is it that I ended up with brown instead of, say, blue? It's sort of a mystery, huh? Don't worry, we're going to get to the bottom of it.
So Gregor Mendel spent a lot of time wondering about his pea plants and how they came to look the way they did. Out of curiosity, he took a purple-flowered pea plant and a white-flowered pea plant and bred them together. So he was sort of expecting to end up with a light purple-colored plant, a cross between the two colors.
But that's not what he got. Instead, all the new flowers turned out the same color purple as the original purple. And there was no sign of the white flower being in the mix at all. So he took the seeds from these purple flowers and planted them, and guess what? In the next generation, he ended up with 3 purple flowers and 1 white. Aha! This led him to make a few conclusions.
First, the law of dominance. For each gene, there are 2 alleles. One of these is dominant over the other. When 2 different alleles end up on the same gene, the dominant one will be the one expressed. That's the law of dominance.
So, in terms of the pea plants, the purple was the dominant allele and the white the recessive allele. Even though the white allele was still present, as its reappearance in the next generation demonstrates, it was being overshadowed by the purple. What a bully.
So let's think about this in terms of sexual reproduction. We've got 2 alleles for each gene, right? That's a pair. Mendel's second law, the law of segregation, says that this pair can separate. In fact, it does separate. After meiosis, each gamete ends up with only 1 allele for each trait. That's so it can combine with an allele from another organism during sexual reproduction.
Say your mom carries 1 allele for brown eye color and 1 for blue eye color. Due to meiosis, each of her eggs will only have 1 of those alleles, so you're only going to get 1 from her. All we're talking about here is the fact that alleles go their own ways into separate gametes. That's all the law of segregation means.
So now Mendel, being ever curious about his pea plants, decided to see if the alleles for 2 traits ever travel together as a package. The answer he came up with was no. Alleles for 2 traits will not necessarily end up at the same gamete. Alleles for each trait go where they go, regardless of anyone else. This is the law of independent assortment.
Mendel discovered this by keeping track of 2 traits at once: seed color, yellow versus green, and seed shape, round versus wrinkled.
He crossed a yellow round plant that had 2 dominant alleles for each trait with a green wrinkly plant that had 2 recessive alleles for each trait. Of course, each plant in the first generation of offspring had 1 dominant allele and 1 recessive one for each trait, having received 1 allele for each trait with each parent. Now, for the second generation, Mendel wasn't sure whether the 2 dominant alleles and 2 recessive alleles would continue to stick together and end up on the same gamete after meiosis, or whether they would all travel separately. What he found is that the pairs did not stay together. He concluded that each allele travels separately, resulting in 16 possible combinations of traits. Don't worry, we'll learn more about these probabilities later.
It's a free-for-all. Every allele for himself, like a game of musical chairs. Except everyone gets a seat in the end, of course. There's no telling what combination of alleles will end up in each gamete.
Are you starting to see why meiosis is so integral to sexual reproduction? It's more than just cutting the chromosome count in half in order to combine with another set of chromosomes. It's also about producing genetically unique offspring.
Inheritance patterns.
Although there's no way to predict exactly which traits offspring will turn out with, thanks to Mendel's laws, we can predict the frequency at which a particular trait will show up.
Whoa, totally sci-fi.
I hope you do not learn too much about predicting genetic patterns in offspring. Otherwise, I will be out of business. But here's some vocabulary to help you along.
Homozygous refers to an organism having identical alleles for a particular gene, that is, 2 dominant alleles or 2 recessive alleles. Heterozygous refers to an organism having 2 different alleles for a particular gene, that is, 1 dominant and 1 recessive allele. Genotype refers to an organism's genetic makeup, what alleles it carries. Genotype of an organism is determined at fertilization. Phenotype refers to how an organism actually looks, that is, what traits it is showing. A Punnett square is a handy diagram used to predict the alleles that will be present in an offspring based on the genetic makeup of the parents. Autosomes are all of the chromosomes except for the sex chromosomes. X and Y are the sex chromosomes. A sex-linked trait is a trait instructed for by a gene found on a sex chromosome.
Okay, let's have a little fun with this. Let's pretend that scientists working deep in the South American rainforest have discovered a new species of tree-dwelling monkey. But this monkey breathes fire. Yeah, these scientists have decided to call this new species the dragon monkey. Oh yeah, did I forget to mention this monkey also has wings?
We'll look at the genes and such of this crazy dragon monkey.
In the dragon monkeys, fire breathing is dominant. The dominant trait is usually signified with a capital letter when we're writing about it. The recessive gene is denoted with a lowercase letter. Most of the time, the letter used is the first letter of the dominant trait.
If you have 2 of the same alleles for a given trait, you are homozygous. Think about it. The prefix homo means same. You may be homozygous dominant or homozygous recessive. If you have 2 different alleles for the same trait, that is, 1 dominant and 1 recessive, then you are heterozygous. Hetero means different.
So, a heterozygous dragon monkey will have 1 fire-breathing allele and 1 non-fire-breathing allele, FF. This is its genotype, or genetic makeup. Since fire-breathing is dominant over non-fire-breathing, this dragon will breathe fire. Yeah, this is its phenotype, what we can see.
And now, we're all ready to do some real genetics problems. That is, let's determine some probabilities for traits in offspring using a tool called a Punnett square. I hope you like squares, because this one is pretty darn useful.
Here we have our dragon monkey parents, Mom and Dad. Mom is homozygous dominant for fire breathing. Dad cannot breathe fire. Since we know that non-fire breathing is recessive, Dad must be homozygous recessive. By the way, this first generation is often denoted the P generation for parental generation.
This is an easy cross. Ready? Mom has 2 dominant alleles, which means 2 capital Fs. It doesn't matter which capital F ends up in the egg. The result is the same. The same with Dad. He's a homozygous recessive, so both his alleles are recessive, and this is signified by 2 small f's. All of Dad dragon monkey's sperm are small f's. When any one of those recessive lowercase f sperm fertilize one of those dominant capital F eggs, the result is the same. All baby monkeys born to this couple will be heterozygous, that is, 1 dominant capital F and 1 lowercase recessive f. That means they will all breathe fire, since the capital F will always dominate. We can conclude that 100% of their offspring will be heterozygous genotypes, and thus phenotypically fire breathers. By the way, this first generation is most often called the F1 generation, for first filial.
Say what?
Okay, let's do another one. Let's follow one of these heterozygous fire-breathing offspring.
Let's say This little one, Esmeralda, meets another little heterozygote, Scooter, and they have a litter of babies. What is the chance that any one of their babies will be a homozygous recessive non-fire breather? Remember, the only way to be a non-fire breather is to have two recessive alleles, aka to be homozygous recessive.
To figure this out in a more organized way, let's do a proper 2x2 Punnett square. Our little dragon, Esmeralda, is heterozygous, so she can contribute either a dominant F or a recessive F to a potential offspring. Our other F1 dragon monkey, Scooter, he can make sperm that also have either a dominant F or a recessive F. So, our Punnett square looks like this. Now we just fill in what happens if this sperm fertilizes this egg, and if this sperm fertilizes this egg.
This Punnett square depicts the chance of each of these four outcomes. Since the homozygous dominant and heterozygous genotypes are all phenotypically fire breathers, there are 3 out of 4 possible offspring that will be fire breathers. Esmeralda and Scooter have a 75% chance of having a fire-breathing baby. To get the percent chance, just divide the top number by the bottom number, just like any fraction, like 3 out of 4. However, there's a 1 in 4 chance that this couple will have a non-fire-breathing baby monkey. That's a 25% chance.
If only the fire-breathing were the recessive trait. I tell you, it's absolutely terrifying. Being alone in the jungle with flames flashing in the darkness all around you, melting your machete into the mud.
Let's recap how we figure out inheritance patterns. First, we figure out what gametes the parents can produce, then separate the gametes using meiosis. List the gametes for each parent on adjacent sides of the Punnett square. Fill in the square, then figure out the percentage chance of each outcome. It's kind of fun. No, really.
Let's move on to another trait, eye color. In our dragon monkeys, purple eye color is dominant to orange eye color. So we use capital P for purple and lowercase p for orange. Say a purple-eyed monkey mates with another purple-eyed monkey. Let's call this the F1 generation, because we also know that each of these monkeys has a parent with orange eyes. What is the chance of an orange-eyed monkey in the F2 generation?
Okay, first, let's figure out the genotypes of the F1 monkeys. Since they have purple eyes, they must have at least one purple dominant allele. However, both F1 generation monkeys have a parent who had orange eyes. Those orange-eyed P generation dragons must have been homozygous recessive, so they must have contributed a recessive lowercase p to the F1 generation. Therefore, both of the F1 monkeys are heterozygous for eye color.
Now that we've done our legwork, we can move forward. Since both F1 monkeys are heterozygotes, what kind of gametes can they produce? Each monkey could contribute a dominant or a recessive allele to the offspring. So, let's list each allele on an adjacent side of a Punnett square. Now, fill in the Punnett square. Remember, there is only one combination that will produce an orange-eyed monkey, pure recessive or 2 lowercase p's. According to our square, there is a 1 in 4 chance or 25% chance that this will occur. Ta-da!
You'll have lots more chances to practice that later and get the hang of it. Let's visit one more inheritance topic today.
Let's talk for a second about chromosomes. All humans have 2 sets of 23 chromosomes each per cell, right? Now, 22 out of 23 of these chromosomes are pretty normal, but that 23rd in each set is special. Those are the sex-determining chromosomes, X and Y. In humans and all mammals and many other animals, females have 2 X chromosomes and males have 1 X and 1 Y.
A sex-linked trait is a trait that lies on one of these 2 chromosomes. Although, FYI, most sex-linked traits in humans are on the X chromosome. Let's try a Punnett square for a sex-linked trait.
In dragon monkeys, as with humans, gender is determined by the X and Y chromosomes. In dragons, long, sharp claws are a sex-linked trait. The recessive trait is short, dull claws. Say a female with short, dull claws mates with a male with long, sharp claws. What are their genotypes, and what percentage of sons would you expect with dull claws?
First, genotypes of the P generation. Since the female has the recessive trait, that means that she must have recessive alleles on both her X chromosomes. So her genotype is X prime, X prime. The prime, that little 1 symbol, is just used to indicate a recessive allele for a sex-linked trait. The male has long, sharp claws, so he must have a normal allele on his X chromosome, and then he has a Y chromosome without any claw Y allele.
Now we make the gametes and do the Punnett square. Both of the mother's alleles are X prime, so we can just write one line of X prime for her. It doesn't matter which of the X chromosomes she contributes, they both have an X prime allele. The father can contribute either a normal X or a Y to the offspring. Fill in the Punnett square. All meaning 100% of these parents' daughters will be XX prime, which means they will have sharp claws but be carriers for the dull claw trait. All the sons will be dull clawed. So the answer to the second question, what percentage of sons will have dull claws, is 100%.
Thanks for watching the show. Take care until next time. Bye.